semester. But tonight we're going to, as I said, delve into the physiology of the respiratory system. And from here to the very end of the course, the story is largely going to focus on how it is that we're able to take oxygen from the air that we breathe and transport it to all the cells of the body. Sounds like a simple story, incredibly complex story. And to talk about this story, you can see down at the bottom there, it says a 5-step model of cellular respiration. That is going to be a model that we begin tonight. It's with us for the rest of the semester. It's a big model, lots and lots of stuff inside of it. But my hope is that for those of you who go off into being a PA, nursing school, physical therapist, pharmacist, you name it, virtually anything that's related to healthcare, this model has an application, one way, shape, form, or another. If you want to go become a ultrasound tech, by the way, I don't know if any of you have been around ultrasound imaging, but pretty darn spectacular. When you sit there and see the imaging that they can do in the heart and all the detail they can see in the heart, you can sit there and think about imaging, ultrasound imaging of fetuses. Really, really impressive. And I'm sure all of you seen those pictures of a pregnant woman. And you know, several weeks out, you can begin to detect the presence of a little child in there. It's really quite amazing. And then when you see fingers and toes, just spectacular. And that's just by sending sound waves, looking at the reflection of those sound waves back. Just that's really, really cool stuff. So we're going to talk about this 5-step model. And it's kind of like the 3 fundamental principles of physiology. And when I talk about this 5-step model of cellular respiration, I'm expecting that you're going to tell me that the first step is known as ventilation. The second step will be gas exchange. The third step will be gas transport. Fourth step, gas exchange. And the 5th step, cellular respiration. I'm going to put a model up there for you in a moment. And you can see that what I'm going to do is I'm going to talk about step number 1 tonight, which is ventilation. And I'll define for you what ventilation is. I'm going to give you a formula that you need to know going forward. We're going to talk about lung volumes. And there are a total of 6 lung volumes that I need you to know. One of which is called tidal volume. That's simply the volume of air per breath. Then there's these other volumes: inspiratory reserve volume, expiratory reserve volume, so-called residual volume, total lung capacity, and vital capacity. That's as far as we're going to get tonight. Once I'm done with that, then you're going to do a lab using the LabScribe system. You're not going to collect any data. I'm going to give you data. The data is actually already out there under course content. But I'll show you how to load that data up, and then your job is to analyze the data. And sadly, you know, there's COVID's had such a huge impact. And one of those impacts is that before, when we used to do this lab, students would breathe through these mouthpieces. But now, of course, we've got all these concerns about somehow contaminating one individual for another, having mouthpieces that are clean, not infected with any viral types of things. So rather than being able to do the lab yourself, I'm just going to give you the data. You'll do the analysis on the data. And what I'm going to explain to you is really the background for that lab. All right. So now that we have clearly established that at least 2 of you were in anatomy last semester, perhaps I'll describe that there were maybe more than 2 of you. be very unfair of me to ask Mr. Cow if there were any other of his friends that were in that anatomy class last semester. So I won't. But I'll find you. And if you don't speak up tonight, when we come back on Monday, I will have done my homework. I will have looked through all the information you provided me at the beginning of this class. Right, Mr. Vargas? So our 5-step model, the cellular respiration, here's how it starts out. Here are 5 steps. What the heck are those steps? Right off the bat, you might be looking at what I've drawn up there going, oh, he's going to talk to us about neurons. This story is not about neurons. This story is about the respiratory system, the cardiovascular system. So that now transitions from the axon terminal. I'm joking about this, but we're going to say that this would represent the lung. What I'm going to show is a logic diagram, not an anatomically correct diagram. Really important for you to appreciate. Does anybody in here want to volunteer that they had anatomy last semester, or do I need to go snooping? Volunteer. As we breathe air in, we're breathing air that's rich in oxygen. As we breathe air out, some of that oxygen's been removed. And we've added CO2. So the air that we breathe out is rich in CO2. Make sure you appreciate that the CO2 content of the air that we breathe, there's very, very, very little CO2 in the air in this room. And yet, the air that comes out of our lungs is relatively rich in CO2. And I've already provided you with why that is. CO2 is a byproduct of cellular respiration. The conversion of pyruvate to acetyl coenzyme A results in the production of 2 molecules of CO2. The Krebs cycle is responsible for producing 4 molecules of CO2. If we're talking about the breakdown of 1 molecule of glucose, right? If you've forgotten all that, go back and look at your notes. The mechanical movement of air into and out of the lungs. The mechanical movement of air into and out of the lungs is step number 1 in this model, and that's called ventilation. So here I'm going to put number 1. I'm just going to put the word ventilation. And clearly, the ventilation rate has to match the rate at which we're using oxygen. If the ventilation rate, if we're not bringing in enough oxygen to match our metabolic rate, then we're in trouble. That's logic 101. What's the definition of ventilation? The mechanical movement of air into and out of the lungs. I want to be clear to help you guys out and make sure none of you make a mistake on the next test. Here's what some students will write after I put that definition up there. They'll sit there and say that ventilation is a mechanical movement of oxygen into and out of the lungs. That's not what's written up there. Ventilation is simply the mechanical movement of air, no matter what its composition is, into and out of the lungs. So please, don't put on the test, don't tell your family member that you're learning about ventilation, and that's the mechanical movement of oxygen in and out of the lungs. It's not. Well, the next thing I need you to know, and this is very, very important, is that the movement of ions, gas molecules, metabolites in and out of the circulatory system only occurs in capillaries. Only. Make sure you have that in your notes. The movement of ions into and out of the circulatory system. The movement of metabolites into and out of the circulatory system. The movement of hydrogen ions into and out of the circulatory system only occurs in capillaries. Why is that? Because the walls of capillaries are incredibly thin. The walls of arteries, arterioles, venules, and veins are very thick. And one of the things that you learn is that diffusion, whenever we talk about diffusion, you have to be thinking about the Fick equation. And one of the components of the Fick equation related to distance is the diffusion distance, right? If the diffusion distance is large, then the diffusion rate is really low. All right. You gotta pay attention, make sure you're catching everything I'm saying. Oh, you don't have to, but to do well in class, I think you do. This is a logic diagram, not anatomically correct diagram. So blood, after it's gone to the tissues, it returns back to the right side of the heart. And it empties into what chamber?
Right ventricle.
The right,
Ventricle.
he said the right atrium. That's exactly correct. The heart's 4 chambers, right? We have the atrium on top, the ventricles on the bottom. But this is not an anatomically correct diagram. This is a logic diagram. So the blood, the venous blood, is coming back to the right side of the heart and emptying into or filling the right atrium. So here I'm just going to put capital R, capital A. There's a valve that regulates the flow of blood from the right atrium into the right ventricle. What's the name of that valve?
Tricuspid valve.
Ooh, thumb up. Very good. Excellent. So here I'm going to put RV. That stands for right ventricle. I'm showing the leaflets of the valve in green. And here I'm going to put, we could put tricuspid valve. I hate these terms. Absolutely hate them. Let me give you what I think is a better term. This is the right side of the heart. So we're going to start out with right. And it's a valve that regulates the flow of blood from the atrium and the ventricle. So we're going to call that valve the right atrium ventricular valve. Or you could call it the right AV valve. And that valve is regulating the flow of blood from the right atrium into the right ventricle. Do we have any other anatomy students from last semester? Mr. Bennett, what's the name of this blood vessel that carries blood from the right ventricle back to the lungs? Fantastic, excellent. So here I'm just going to put capital P, capital A. That's our pulmonary artery. So I'm just going to put an arrow here so that we're showing some directionality. All of you learned this probably in our first week, maybe the second week of class. But oxygen is going to flow from the lungs into, what's the name of the blood vessels here? Eric, what would you tell me? Okay, all right. You said pulmonary vein. Do we have any other anatomy students from last semester? It's amazing how many we're uncovering.
Pulmonary capillaries.
2 thumbs up, that's exactly right. Remember, the diffusion only occurs at the level of capillaries. So it can't be the pulmonary vein. It has to be the pulmonary capillaries. So whenever you think about something moving back and forth from the circulatory system into a cell, vice versa, that can only happen at the level of capillaries because that's where the walls of the vessels are very, very thin. Okay? So it's just a simple rule. Will help you out immensely. So I'm gonna come along and I'm gonna put up here that along this segment here, we'll just say that this represents our pulmonary capillaries. So as the blood flows past the lungs, it flows past the alveoli, it becomes oxygenated. And so now I'm just going to represent that by showing O2 moving into the pulmonary capillary region, and I'm going to show CO2 leaving the blood. And now this oxygenated blood flows from the lungs towards the left side of the heart. And what's the name of the blood vessel that's carrying this oxygen-rich blood back to the heart? Anybody?
Pulmonary vein.
Pulmonary veins, right? Because the definition of an artery is a blood vessel that's bringing blood away from the heart or towards the heart.
Veins towards.
Arteries are away from the heart and veins are towards the heart, right? Pulmonary artery, away from the heart. Over here, pulmonary vein. Here I'm going to put PV. So the blood from the pulmonary vein then is eventually carried back to the left side of the heart, and it empties into the, this is logic 101. It empties into what? The right ventricle, right? Right atrium? Left atrium? Left ventricle? See, logic 101 is over here, we've got the right ventricle on top, so the left ventricle has to be on top over here. What do you think, Mr. Lamb, is that correct?
Left atrium.
It is the left atrium, that's exactly right, very good. And yes, you were right too. So here we've got the left atrium. Well, the blood then is pumped from the left atrium into the left ventricle. Notice how I'm showing the direction of the leaflets. Got to make sure you understand that, because that's part of the logic. Well, from the left ventricle, the blood is pumped out into what major blood vessel? Anybody? So from the left ventricle, the blood is pumped out into the aorta, right? To the aorta. Here I'm just going to write aorta. And then from the aorta into the arterial tree, into the arterioles, and then from the arterioles into what, Mr. Corey? That's exactly right. Very good. So here's our cells. I'm just drawing a little black box down here. And that black box, we're going to say, is a cell. But really what I want you to do is interpret that little black box as being all of the cells of the body. So the blood is pumped out of the left ventricle, out into the aorta, into the arterial tree, and then into the capillaries of the tissue bed. And here we have the diffusion of oxygen out of the circulatory system into the cell, and that oxygen diffuses into the mitochondria of the cells. You also know that CO2 is a byproduct of cellular respiration. So CO2 is produced by the mitochondria. Okay, the 5-step model of cellular respiration. Here's the model. Let me provide the other parts of the model. Step number 1, ventilation. What comes after step number 1? The answer is step number 2. What is step number 2? It's called gas exchange. Right now, underneath gas exchange, I'm going to write Fick's law of diffusion. oxygen enters into the circulatory system. Okay, great. But we got to get that oxygen transported to the cells of the body that need it. And that's step number 3. Step number 3 is called gas transport. I need 2 main factors to determine gas transport anyway. Well, that's under one of the 2 key determinants. What's creating blood pressure? All right. Anybody else? Cardiac output. If your cardiac output is 0, then what's gas transport? What's the delivery of oxygen to cells of the body? 0. Here I'm going to put cardiac output, and beneath that I'm going to put Q dot. What does a dot above any symbol mean? Say it loud. Yeah. It's a rate, some type of a rate, right? Q dot means the rate of blood flow, or the rate of any other flow you want to talk about. Well, that's one important factor. What's the second important factor? We've got a pump. That's all under cardiac output. That's a good point. What else? Don't be bashful. What else do we need to be able to transport oxygen? Okay. The answer I'm looking for starts with an H, ends with an N. Hemoglobin, right? Because guess what? The solubility of oxygen in water, which is the majority of our blood, is muy poquito, very small. So we have to have a molecule to help transport. You guys learned about carrier proteins, right? Well, here's another carrier protein, except here we're not talking about endocrine type of function. We're talking about the ability to capture a molecule and transport. So here, under gas transport, I'm going to put the O2 content of blood. And you'll learn that one of the main determinants of the oxygen content of blood is concentration in blood. Okay, so that's step 3. We got 2 more steps to go. What is step 4? And the answer is step 4 is gas exchange. Except now we're talking about gas exchange at the tissue level, not at the pulmonary level. And you guys already have the answer. What determines gas exchange at the tissue level? Anybody? Say loud. Come on, please. Fixed law of diffusion, right? Very good. Here's a cool thing. You guys have already learned about diffusion. That's step 2, step 4. And you've already learned about step 5. What is step 5? Cellular respiration. Anybody give me a definition of cellular respiration? Don't be bashful. Go for it. Okay.
The use of oxygen as a final electron receptor.
The use of oxygen to produce ATP. 2 thumbs up. That's great. That's awesome. Very good. There's your 5-step model. You're not going to believe how much stuff fits into this model. There's a lot. This is the rest of our semester. Okay. So, yes, go for it.
That boric acid exchange, that tissue, right?
That's correct. So that's occurring in the pulmonary, that's in the capillaries of the tissue. Well, I'm just going to say that, our evolutionary history, you have to be amazed at human evolution. As I try to explain to you, we are miracles. And how the heck is it that we can sit there and launch a 450-foot rocket and have the darn thing come back and land it with precision and catch it before it touches the ground? That's mind-blowing. That's a lot of incredible physics right there. Absolutely amazing stuff. But sit there and think about 500 years ago, say in the 1500s. You had people like Galileo, you had people who were looking at the stars, they didn't know what the heck was going on out there. Sit there and think about the whole idea about the Earth being the center of our solar system. That was like the prevailing thought way back then. And now we know so much more. But imagine yourself being a human biologist in the year 1700. You didn't know any of this stuff. But you sat there and you're curious and you're sitting going, you know what? I noticed that people are breathing all the time. That's got to be important. I think I'm going to cut open a corpse and I'm going to look at the lung. You do that, you look at the end and go, well, this is pretty cool. And then you sit there and go, you know what? I'd like to actually measure how much air goes into the lung during each breath. And so you begin to make these types of measurements. And these measurements are referred to as spirometry. Spirometry, the measure of lung volume. So you sit there and you go, you know what? The lung is a pump, and I want to study the pumping action of the lung. So we're going to create a graph, and on this graph we're going to have volume on the y-axis, and this is going to be in the units of liters of air. And then we'll say on the x-axis we're just looking at time. And we'll just say that this is in units of seconds. So these ancient physiologists sat there and they developed these devices to measure lung volumes. They simply would have people breathe into devices where they can measure the volume of air. Spirometry. The first thing I want you to recognize, a very simplistic representation. So this goes under step number 1 up there. Very, very simple representation of the structure of the lung. Well, these early physiologists, early anatomists, they looked at the lungs and they observed that the trachea was relatively rigid. And there were these branching airways coming off of the trachea. And these were the bronchi. And then there were branches coming off the bronchi. We'll refer to those as bronchioles. And at the end of this, there were these little sacs, these little inflatable sacs that all of you would tell me are referred to as alveoli. So you sit there and you look at the trachea, the bronchi, and the bronchioles, and you're sitting there going, these are pretty rigid structures. So that when people breathe in, it's not the dimensions of the trachea or the bronchi or the bronchioles that are changing. They're rigid. It's the inflation of the alveoli. And the movement of air out of the lungs is the deflation of the alveoli. So all of you would tell me this, all of you know this, is that during inhalation, the alveoli inflate. And then during deflation, they get smaller. And this cyclic event that occurs over and over and over. Now, why am I putting this up here? The reason, or the answer to my question, is that you can breathe out as much air as you possibly can. Here's Logic 101. Is there air still in the lungs? You breathe out as much air as possible. Is there still air left in the lungs? It's really a simple question, Logic 101. What's your answer? Yes or no? It's definitely a yes, right? Because we've got these non-collapsible structures. And I'm hoping in your anatomy class that you learn that there's this term to describe this space, and it's referred to as anatomical dead space. How many of you have heard that term? Anybody? Nobody. Anatomical dead space. Why is that important? Because in this graph, we're not starting out at 0 liters. There's never 0 liters of air in the lung. Everybody with me on that or not? Confusing? Hopefully not. So here I'm going to start out at the bottom. I'm going to put 0. And then I'm going to go up in increments of 1 liter. So going back to our ancient physiologists, anatomists, they've got a live person, they've got a mouthpiece in their mouth, there's a hose, and this goes into a device that allows them to see the volume of air that comes out and goes back into the lungs. That's called spirometry. So they see something that looks maybe like this. thinking logically, look here during this phase. What's happening to the volume of air in the lungs? Please. What's happening to the volume of air in the lungs during that phase? It's going up. That's what? Is that expiration or inspiration? It's inspiration, right? And of course, you would tell me that the opposite represents expiration. How would I actually measure the volume of air per breath? The volume of air per breath is simply the difference between the top of the peak and the bottom. And that's the volume of air per breath. Let me give you a name for that. But before I do that, again, we're going back to ancient times. Yeah, 300 years ago. It's not ancient times, but it is far removed from where we're at. These folks, they knew about the ocean, and they knew that the tide would come in, and the tide would go out. And they sat there and said, you know what? The movement of air into the lungs and out of the lungs is cyclic, like the movement of the ocean. And so they came along and said that we're going to call this measurement tidal volume. I don't like the term, but it is what it is. I would just call it the volume of air per breath. But how scientific would that be? So here, what's our abbreviation for tidal volume? Well, we're in chemistry land. What's our abbreviation for volume? All you can tell me, capital V, right? Capital V, lowercase capital T. You're going to read that as tidal volume. What is tidal volume? It's simple. It's the volume per breath. That's what it is. Now this measurement is typically made under resting conditions. So you have your subject, they're sitting there, they're in a nice quiet environment, you've got them hooked up to a spirometer, and they're just breathing normally, just as all of you are doing right now. Another part of this measurement of lung volumes is to ask the subject, after they've done this resting period breathing, is to suddenly try to breathe in as much air as they possibly can. What are we going to call this this measurement? Again, I don't like these terms, but this is what the community uses. That's what we go with. This is called the inspiratory reserve volume, ERV. So here I'm going to put Well, if you can ask your subject to breathe in as much air as possible, you might sit there and say, okay, now I want you to try to breathe out as much air as possible. And that, in fact, is what happens during these spirometry tests. And this volume is known as the expiratory reserve volume, the other herb. Hopefully you'll notice that I did not show the value going down to 0. That small volume that's left over, that's our dead space. We cannot ventilate that. We can ventilate it, but there's no gas exchange occurring there. And that's our residual volume. And that's simply referred to as RV, the residual volume. So far I've described 4 lung volume measurements. There's 2 more. We're going to talk about what's referred to as vital capacity. Vital capacity I'm going to put VC. VC is simply the sum of IRV plus tidal volume plus ERV. That's vital capacity. And then there's what's referred to as total lung capacity. TLC. And TLC is the sum of IRV plus tidal volume plus ERV plus RV, the residual volume. Okay, questions? Dr. Dash will speak up. Yes?
How do you calculate
Good question. How do you actually determine residual volume? And to do that, you use nitrogen, or you can use helium. Washout techniques, these are fairly sophisticated techniques. You need sophisticated gas analyzers to do that. So typically what happens is that you take all this data, there are large databases in terms of normal values, you put that in there, you come up with, if you know a person's sex, their weight, their height, you fit that data in there and it will help predict what the residual volume is going to be. It's kind of predictive. Hard to actually measure Not impossible, you can do it, but you just have to spend a lot of money on your equipment to do it. Let me add one other thing. So coming back here to ventilation, the rate of ventilation is incredibly important. So up here, we're going to put a formula in for the ventilation rate. And here's the formula that we're going to put. I'm going to put capital V, that stands for volume. Then I'm going to put a dot above that. You're going to read this as ventilation rate. And the ventilation rate is equal to the tidal volume times breathing frequency. Breathing frequency is described as F subscript V. I'll have more to say about that. We'll actually do some calculations on ventilation rates. Let me say though, that if I were to ask any one of you to run around this building, what would happen to your tidal volume? Ms. Kaylee, what would you say? What would happen to your tidal volume?
Increase?
What would that mean though? I mean, when all of you start running, if you go out and run for several minutes, you're going to sit there and notice that you're starting to take deeper breaths, right? So your tidal volume definitely goes up. And not only does your tidal volume go up, but what happens in breathing frequency? It goes up as well. So one of the points that I want to make is that please understand that tidal volume is simply defined as the volume of air per breath. And that depends upon what our physiological state is. If we're in a resting state, then the tidal volume is relatively small. But if we start to exercise, now we breathe in more and we breathe out more. And the tidal volume increases. We're not changing the maximum amount of air that can come in the lungs. We're not changing the maximum amount of air we can get out of the lungs. We're changing the volume of air per breath. So some people sit there and say, oh yeah, I breathe in more, but I still have this inspiratory reserve volume. No, we're starting to eat into that inspiratory reserve volume so that we can increase the volume of air per breath. So the definition of tidal volume is the volume of air per breath. I don't care what condition we're talking about. All right, so now all of this is a prelude to what you're going to do tonight. If you go out to the course content, let me just get there. All right, if you go out to the course content for the class, and you look at these two files here, there's a one that says spirometer lung volumes. That's the actual data. And then there's this PDF file which describes the analyses that you need to perform on them. So I'm going to come out here and I'm simply going to go over to LabScribe. Everybody sees that hopefully up there in the top left-hand corner. So I'm going to start that. And then it's going to ask you for hardware dialog. Just say OK. And then it'll ask about the wire device. Just say OK to that. And then you're going to go up in the top right-hand corner. And you're going to load up the data file. So I'm going to come out here. I'm going to go to Open. And let's see here. Where did I put my data? And you're going to click on that data file, and you will see two channels. You'll see the channel at the top. That's the raw data. You're going to ignore that. The data that we're after is in that second channel on the bottom. So here's the spirometry data. You can see that the units are in liters. And you can see that there's this rhythmic breathing pattern here by this individual. And I'm not going to go through all the instructions, but what you're going to do is you're going to come along and measure the heights of these, and that will then give you your tidal volume. We come over here, and you ask your subject to breathe in as much air as possible. The difference between the peaks of the normal breathing and this maximal breath in, that's the inspiratory reserve volume. In the same maneuver, you ask your subject to breathe out as much air as possible. The difference between the bottom of the resting breathing pattern and here is the expiratory reserve volume. This first set of data, this is important for you to realize, that's under resting conditions. The second set of data is following exercise conditions. And I just want to alert you to the fact, take a look at the deflection here per breath, and you'll notice that the deflections over there are significantly larger than they are over here. Why? Because during exercise, the muscles demand more oxygen be delivered. So you have to increase the ventilation rate to satisfy the metabolic needs of the muscles. So this is the data you're going to analyze. You're going to open up that PDF file that's also posted out there. And let's just see here. And the PDF file is this one here. Again, you're not hooking up any subjects, you're just using this data. Try that again. There you go. So you can see here they're showing you the measurements you're going to make for tidal volume, inspiratory reserve volume, expiratory reserve volume. They've got some other measurements in here that you need to make as well. And you've got two conditions, resting and exercise. You're going to answer the questions that relate to the resting and the exercise conditions. I knew it was going to do that to me. Oops. And you've got some tables you need to fill out. Here's some reference data for you. Yeah, I can't scroll to it. But you'll see the tables down below. I'll help you out. If you've got questions, I'll come around and help you out. But to do this, you gotta first load up LabScribe on the computer, and then you can access that data and make the appropriate measurements. So with that, get the show on the road. Table number 2. Table 4. 4. Eric. Table 5. Table 6.
some classes this semester. Actually, I didn't get a. The only reason why I knew to look at my grade was because I took the first one. Yeah. And then I said, do they know those two days?
Oh yeah, he changed the grade for example.
Oh,
At least for
okay.
mine.
Okay, because it still needs to change for mine too.
Oh,
It says about
Yeah, it happened to me too, but he changed it so like
Okay, also.
do you remember the one for the hypercalcemia?
Yeah.
Yeah, for
Did you put high calories? I put high calories, but I think that was wrong. That's why I got it wrong. Yeah.
the one for increasing calories.
Elevated calorie.
I selected that one. I don't know why it's wrong because technically it can be perceived as like increasing.
High calorie.
Yeah, because kalemia means potassium in blood, so increasing potassium in blood is hypercalcemia. Wait, technically no, because hypercalcemia means like abnormal, or just increasing potassium in blood. I don't know.
No, but that is, yeah, hyper. You're abnormally high.
Yeah,
Because you wouldn't say that it's high if it wasn't abnormal.
yeah, but I mean, I guess if you say like increase in potassium in blood doesn't necessarily mean like hypercalcemia. Maybe just like an increase. That's why I think it could be wrong, but I think it should be like I don't know.
Wait, metformin.
Metformin is
It causes. Wait, does it cause? It causes glucose levels.
to rise, or does it?
I said what reduces glucose. It causes it to rise.
I think it's used for people with hypoglycemia.
No, it's for people with diabetes.
Class, I should have mentioned you're doing exercise 1 and 2. There's 3
Low
exercises.
is low.
You're not gonna do the third one.
So what is
Hyper
metformin?
is high.
Yeah.
Metformin, it's supposed to